So good morning. I'm very excited to be here today to tell you how we're making drug development faster and new drugs safer. I'm going to be telling you about our proven technology, which is covered with IP. We've got existing demand, we're ready to scale, and we're poised for growth. So your stomach feels a little unsettled. You feel bloated. You take Cybercyte, and you expect your digestive system will settle down. What you don't expect is a heart attack. Cybercyte was generating $1 billion a year in sales for Johnson & Johnson before it was withdrawn from the market from the FDA, following 18,000 cardiac events and 302 deaths. In fact, a wide range of non-cardiac drugs result in heart attacks and death, including drugs designed to treat cancer, infection, allergies, malaria, all kinds of drugs. Following several high-profile, high-cost withdrawals, the FDA mandated that every single drug that comes to market must demonstrate that it doesn't disrupt the electrical activity of the heart, and specifically that it doesn't cause heart attacks. This is known as the FDA cardiac safety screening. Now, it's a $9.9 billion global market. And it protects patients from cardiac arrhythmias and death, but it's also the point at which 70% of drugs fail and fall out of the drug development pipeline. So let's take a look at that drug development pipeline. You start with 5,000 to 10,000 compounds, and about 250 show promise and undergo preclinical testing. 5 will make it through to clinical trials, and only 1 will make it to market. It costs on average $1.3 billion to bring just one drug to market. 249 of the drugs in the preclinical phase won't make it to market. That's an awful lot of time, effort, and money being spent on drugs which will ultimately fail. The key to making the drug development pipeline cheaper and faster is eliminating bad drugs more rapidly. The current standard test for FDA screening is something known as the hERG test. It occurs early in the preclinical phase, and it's a key decision point about whether to go forward with preclinical testing. The trouble with the hERG test is it's wrong 30% of the time. Despite this, it's a $774 million per year market in the US alone. Drug safety screening and hERG testing isn't performed in-house by pharma or biotech companies, but it's farmed out to contract research organizations or CROs. Cytocybernetics is just such a CRO with the distinct advantage of our Cybercyte assay, which is 4 times faster and twice as accurate as the current testing mechanisms. We eliminate bad drugs from the pipeline earlier. Early and more accurate testing results in savings of hundreds of millions of dollars for each drug brought to market. Our competitive advantage in this field is the technology behind the Cybercyte test. We combine cells, computers, mathematical modeling to turn noisy signals into clear, accurate, and reliable signals which are suitable for rigorous quantitative analysis. The result is an enormous improvement in the predictive ability of our test compared to our competitors. We have 2 market segments. We're selling the technology to R&D. We've already sold 5 units and anticipate garner $2 million a year by 2020. We're also performing drug safety screening for pharma and biotech. We anticipate $30 million a year by 2020 on a very conservative estimate of garnering 4% of the US domestic market. We already have 3 contracts pending. Our technology is going to disrupt drug safety testing, and our technology is a major R&D breakthrough. That's not just my opinion. We've got external validation. We were recently awarded an NIH Small Business grant, and we were given the perfect and very rare score of 10, the highest possible score. The These anonymous reviewers were very positive about our technology. They said we had great potential to disrupt the way drug safety is evaluated and will significantly impact the field. We're also awarded the prestigious SUNY Technology Accelerator Fund Award, and the part of the due diligence was sent out to a drug company executive. He liked our technology so much, he reached out to us. We're finalizing a contract with him. We also have New York State UB-CAT funds, and we were one of the 12 bright ideas in the National Pharmaceutical Congress. We've assembled a world-class team with over 40 years of cardiac safety testing CRO experience. Randy and I have several previous inventions, including a device to treat postpartum hemorrhage in low-resource settings, which the Gates Foundation invested $100,000 in. We're particularly excited to have 2 buffalo boomerangs. Shimin comes back to us from a Seattle biotech, and Mark we recruited back to Buffalo from Silicon Valley. Our lead scientific advisor is Dr. Gary Rasmussen, who led a medicinal chemistry team at Merck. He has 45 patents, including those for Proscar and Propecia. Our market size is $9.9 billion a year globally, but there are only about 100 drug safety testing CROs worldwide. This reflects the enormous barrier, both intellectual and financial, to entering this market space. Our patent-pending Cybercyte test is 4 times faster and twice as accurate, which is going to save hundreds of millions for each drug. We're finalizing contracts with 3 companies. We've got the additional market of the direct device sales. We've got worldwide sales, US, UK, and Singapore. This is Randy with our very happy customer in Singapore. He's going back out there to install another system next year. The device sales, although small, fuel the screening services. Our partners are providing external validation by publishing their work in leading cardiac journals and essentially free advertising. We've never advertised, but we have customers and contracts. We've invested $473,000 in grants and personal funds into this company. We're now looking to raise $3 million in new investments. This will enable us to scale our technology and our service capabilities. We're going to invest in high-throughput screening equipment. This will enable us to enter the high-throughput screening market, as well as to scale our technology and provide additional IP, which will then validate and beta test with our pharma partners. We're also going to invest in people. We're going to recruit key business development and an executive team, as well as skilled operators, computer programmers, and support staff, and of course, an advisory board. We have the investment from 43North. We're also well positioned for the Phase 2 Small Business Award, given that we got a perfect score in the Phase 1, and we've met all our milestones. We're raising additional funds from angel investors and VCs, as well as our strategic partners. Those discussions are ongoing. This is our projected sales and growth based on our very conservative estimate of 4% of the US market. Device sales, although small, will keep us cash positive in the early years as the screening services expand. The key factors driving growth will be the publication of our current R&D work, including the validation that we are 4 times faster and twice as accurate, and also the investment in the high-throughput screening services. We're going to be creating some great jobs in the Buffalo biotech corridor. We'll create 5 jobs in 2016, 20 in 2018, and 40 in 2020. These are high-tech jobs, highly skilled, $30 to $50 an hour. So I've shown you the proven technology. We're covered by IP, and we've got existing demand. We're ready to scale. We're poised for growth. We're really excited to be bringing drugs to market faster and to be saving lives. Thank you very much for listening, and we welcome your questions.
I really got to get to North Tonawanda. I had no idea that the accent in that North Tonawanda has changed so much. I have to go there a little more.
Oh, no. no, it's not changed. We've been there since 1812.
Oh, I missed it. I'm sorry.
There's a little enclave left over from 1812 war.
Lycara, it's all yours.
Yeah, that's okay. Can you? Yeah, you can hear me.
Yep.
So I read over some of what I've looked at this. How do you think this overcomes like what you can do with like induced pluripotent cells, right? If I'm making a card, I know you have an algorithm for it to get around the electrical conductivity. So I'd like to understand that because that's actually growing. And I'm referring to more or less CDI and what they're doing with Bob Pillay and that group.
Absolutely. So the technical explanation is we're using induced pluripotent stem cell-derived cardiomyocytes as part of our predictive test.
Okay.
The problem with these, excuse the technical explanation,
Sorry.
we left it out for the business model, but the induced pluripotent stem cell-derived cardiac myocytes are missing a key electrical component. This is the IK1 electrical component, and some groups have tried to put that back biologically. It doesn't work. So cardiac cells, they've got automaticity, they're not the real thing. What we have done is we've got a mathematical model of the IK1 current, which we couple in real time to the iPSCs. Great. So that's our technology, is making sure that we can mathematically determine what the current is and replace it in real time. The cells are beautiful. I would show you action potentials. I would love to go into that with you.
No, we won't. I was just curious about that because I've
So that's our
dealt with
market.
them a fair
The
amount.
iPSCs, it's fantastic new technology. It's only about 5 years old. These are available all over the place, and it's amazing, and they're almost there, but they're not quite there. So we use the R&D people, the iPSC people who are using our technology for all kinds of R&D as well.
And as part of your model, and I'm going to let others jump in here, you're going to sell to R&D and then maybe for the big players, and then you're going to do maybe outsource and do the service yourself for the smaller biotechs.
So it's interesting.
I'm trying to understand. Yeah.
The people who want to buy the technology directly are the people who are focused on R&D.
Right.
It's got a whole extra market in personalized medicine, or as now precision medicine, because you can do it patient-specific with iPSCs. There's a whole market out there. The drug companies have told us they don't, it's such a highly skilled technical operation.
That's what,
They don't want to be doing this in-house.
Okay.
They want us to do the work for us. They want to ship us the drugs. They want to have the analysis done, and they want the answer. And so that's what we're doing with pharma, is the CRO. That's why we've got that model. Yes?
Well, I have a question. If the opportunity is really in testing, as it shows in your model, why are you using precious resources to be in the selling of the machines? Why aren't you just focusing solely on the testing side?
So the device sales are relatively small, 2 million a year by 2020 versus 30 million a year. But these are key opinion leaders. What you have to do is validate your technology and demonstrate how superior it is. All of the 5 people we've sold the device to, they've published in leading cardiac journals. The demand is just increasing when people see what the technology can do and validate it. So it's worth putting the effort into the device sales, because that's how you, excuse me?
It's your marketing.
So wouldn't you just give them away for free? I mean, it doesn't sound like a selling business, but more.
With the academic units, yes, we have a relationship with academic departments to further their research. It's they pay. I'm not going to turn down their money. Yes.
So talk about how good you need to be, and how is your secret sauce been validated, tested from a regulatory approval standpoint, and is there any other technology, even in sort of the research lab phase, any papers that are coming out in terms of things that are potentially threats or competing with what you're doing?
Well, that's quite a few questions, so I'll sort of work backwards. No, there are no threats really occurring at this time. We know quite a bit about it because we both spend a lot of time on peer review for the AHA and the NIH, as well as serving on numerous editorial boards. But we do anticipate that sooner or later that IK1 issue is going to be sorted out. But problem still remains, and the visit still remains a viable method because this is a good way of interrogating cells much more effectively to extract maximal information. And also, our specialty is, of course, in the signal processing and interpreting of the data that's coming out, both of which aren't going to go away as technology advances, and we're in a key position to do that.
Okay. Validation, regulatory approval?
We're not subject to regulatory approval because this is a very early stage. We have to do some good laboratory practices of validation, which Shimin Wang has been through now 3 times through 3 separate buyouts in his CRO that he came to before coming to us. So we have to do that, but by and large, this is not in human patients, so we don't have any barriers regulatory-wise towards getting this done. We just have to be careful and maintain good laboratory practices.
Wait, but still don't you need 510K and CE?
Right, but the FDA doesn't validate any specific assay. And in fact, because of the 1.3 billion of bringing the drug to market, the drug companies went to the FDA and they're asking for some new technologies. So they came out with a comprehensive in vitro proarrhythmic assay, or the CEPA initiative, where they're looking for combining iPSCs, mathematical modeling to have a better test.
Yeah, so you're going to use this on the research side initially for basically, it's going to be drug screening essentially. If you want to go to 510K route and monitor down the road if someone is taking a drug, if there's something later, you could do that. That's more of a test. It's not what you're trying to do now.
Absolutely.
What you're doing now is on a so I have a question. You said you're going to be 2 times more accurate, so I'm thinking maybe you're going to come in at 15% at a hERG failure rate. What I'm trying to figure out is, okay, what are you capturing that these other tests are missing? Because they used to, you do tests for QT prolongation, those kind of things. That's what I want to understand to see how you're going to get to that.
So when you're spending 1.3 billion, the 15% would translate into substantial saving.
Right.
And we actually took the 15% of the current best assay, which
Right.
would be using iPSCs
Right.
without our technology. We're enormously more accurate than a hERG.
Right, that's what I was figuring out, like, how did you get to
Enormously
that?
better
Because
than
the
hERG test.
the hERG
But
test
it's-
has been out there for a while
Yes.
on this, so how do you've extrapolated based on what's been missed?
Right.
Exactly. It's tough.
So
Sorry.
we did it through an analysis of the variance that you get going from an assay without our system in the induced pluripotent stem cells versus one with our device. And so you reduce, as you see that signal, it goes from messy to clean. That's where we come up with half the variance, so that's the factor of 2, and then 4 times because your power goes up as the square of your
Right.
variance.
Yep.
So, but there are other ways we could arrive at better assays because we're comparing apples to apples, but when we're looking at current methods, really, which are hERG, we're comparing apples to oranges.
Right, because I ask, because you can look at there's been enough cardiotox failures, right, in phase 3 or whatever, that you can look at and actually see, okay, what was missed or whatever, and go back and use this.
The CEPA initiative has a panel of drugs,
Right.
and they say that anything that you're going to come up with has to be tested against that panel of drugs. And we're working our way through that panel of drugs right now, and that's what part of the investment is going to be.
All right.
And you had a question?
Yeah, so I'm not going to talk about the science.
Yes.
I don't know the science. But this is a business plan competition.
Yes.
So we're not judging you based on necessarily the size of the business. We're judging you based on the ability to really, be leaders and take this idea forward. I have a lot of confidence in the science of this. I don't know if that's kind of grounded in anything. I have less confidence in you two as kind of, on the business side. I look at the projections, it seems like blunt instruments. I look at the manufacturers of these, the cells that you're using. They have distribution, how do you work with them to get it out? And so talk about who in your team is going to really grow the business and be savvy, because it's not an easy market on the business side.
Right. Initially, we're using the connections we already have within pharma, people who've contacted us and such.
But who on the
to grow by-
team is going to grow the business, not the science?
Well,
You guys have that
we
covered.
do need some key business people. That was a key portion of our business plan. that's one of the things we need to do with this
So you don't
money
have
is to
anyone now?
grow it-
Oh, we
grow it.
do. Our core team has got 40 years experience in specifically the CRO market.
But they're not
Shimon has
necessarily-
had 3 exits already. So he's very familiar
And who
with
is that?
Shimon Wang.
Okay.
He's had 3 exits. We recruited. We're very excited to get him back from his
He's a
latest-
boomerang.
Thank you
He's
very
a
much.
Buffalo boomerang.
One of the
We're
boomerangs.
really proud of the boomerang.
Thank you very much.
Thank you.
Appreciate it.